everyone, this is Jonathan Lovas speaking. I'm a former graduate student of the Yuste laboratory at Columbia University, and today I'm going to talk to you about something amazing that happens to Hydra. You can dissociate it into individual cells, and under the right conditions, it puts itself back together. Yes, you heard well, the animal can reassemble itself from individual dissociated cells. This happens spontaneously, and for my thesis project I studied how patterns of activity of the nervous system change as the animal regenerates. Ultimately, we observed that neurons synchronize spontaneously into small ensembles, which begin interacting and form larger and larger groups until they all work together. But let me start at the beginning. Hydra is a freshwater cnidarian, and as such, is a relative to jellyfish, corals, and sea anemone. Cnidarians are ancient. It's thought that they diverged from bilaterians 750 million years ago, and they arguably have the simplest nervous systems in evolution, which is one of the reasons they're ideal to study. For almost 300 years, however, the amazing regenerative abilities of Hydra have captured the fascination of scientists. Although Hydra was originally described by Leeuwenhoek on some of the first microscopes, the first experiments on Hydra regeneration were conducted by Abraham Tremblay in 1744, where, much to his surprise, the animal showed the very plant-like characteristic of regeneration, much like the mythical creature. Much later, in the winter of 1896, Florence Peebles, under the supervision of T.H. Morgan in our department at Columbia University, began a series of experiments that advanced the study of Hydra regeneration. She determined the minimum size of a Hydra fragment capable of regenerating into an animal. Pieces of Hydra roughly 1/6 of a millimeter in diameter will regenerate, yielding hydrants with 1 or 2 tentacles, never more than 2. While fragments smaller than this disintegrated after a variable amount of time in isolation, it was later observed that these smaller tissue fragments, even dissociated cells, exhibited their own interesting behavior in the presence of additional tissue fragments. Not long after the introduction of the first immortalized cell lines, efforts to culture Hydra cells began. Numerous attempts in the 1950s and 60s were able to maintain viable cells in culture for upwards of 1 week, but the majority of cells in culture would not divide. Only the nematoblast population that gives rise to the animal stinging cnidocytes would proliferate. Importantly, however, these efforts discovered 2 vital pieces of information that ultimately allowed the reaggregation and then regeneration of intact animals from individual dissociated cells. The fact that dissociated cells require media of much higher osmolarity than standard Hydra media to remain and survive in isolation, and the fact that this higher osmolarity media makes the freshwater Hydra fragile and susceptible to relatively gentler means of mechanical dissociation to isolate cells. Geyer et al.'s 1972 publication reporting the regeneration of Hydra from reaggregated cells marks a milestone achievement in the field with the establishment of a protocol for the regeneration of intact animals following dissociation into individual cells. Geyer's work revealed several interesting aspects of the phenomenon. Notably, the work confirms that Hydra regeneration is based predominantly on morphollaxis, the rearrangement of cells to form new tissue, as opposed to the epimorphosis-based regeneration of vertebrates, in which undifferentiated tissue forms a blastema and repatterns missing tissue. Today, the application of modern imaging technique in neurobiology allows interrogation of the animal's nervous system in unprecedented detail. Here, we see the pioneering work of Dupre and Yuste, which uses the calcium indicator G-count expressed in every neuron to detail the activity of the non-overlapping nerve nets of Hydra vulgaris. As the focus of this work in current biology, we use calcium imaging to visualize Hydra's nervous system to understand the amazing regenerative abilities of Hydra, and we leverage this ability to observe the complete functional development of a nervous system. Here, we image the neuronal activity of regenerating aggregates of dissociated Hydra cells at 8 hours, 24 hours, 48 hours, and 72 hours. We were first intrigued that at 72 hours, the patterns of the alternating firing of rhythmic potentials and contraction bursts across the entire animal appeared to resemble the dynamics of the intact animal. After analysis, we concluded that by 72 hours, Hydra's nervous system was largely synchronized, and we focused our analysis on earlier time points. With extensive tracking of individual cells, we were able to extract the firing patterns of individual neurons to observe how they change from time point to time point. Comparing the correlation in activity to the distance between pairs of neurons, we observed that strengthening appeared to occur first at short distances until 48 hours. This suggested a potential role for modularity in the process, so we then used the popular Louvain algorithm to partition the network into modules. In the case of the developing nervous system, these are neuronal ensembles that form during the process. Moreover, with additional analysis, we were able to detail the hierarchical structure in the developing ensembles. We next explored how these modules interact. One interesting feature of the process of reaggregation are the waves of activity that spread across the animal. Looking at the distribution of the sizes of these waves, we were surprised to see dynamics reminiscent of the constitutive function of the mammalian cortex, which has been suggested to operate at the state of self-organized criticality, a concept used to describe dynamics of systems attracted to a state just at the verge of a phase transition. Intrigued by this, we next explored how our detected modules relate to these waves of activity. We found that, as Hydra's nervous system synchronizes, these smaller ensembles begin interacting with each other at different rates before global activation across the entire animal. In conclusion, we found a role for hierarchical modularity in the reassembly of Hydra's nervous system. How do you build a nervous system from scratch? Module by module, just like you would build a house. Except that here there's no architect giving instructions. Somehow all neurons know exactly what to do. We don't understand how, by the way. This is just the beginning of our journey, and the self-assembly is just one of the types of fascinating questions you can ask with Hydra. Thank you for your attention, and we hope you'll take a closer look at the paper and email us if you have questions.